All right, so today we're diving into drug discovery.
And more specifically, how scientists actually validate a target.
Yeah, it's like, before you build a house, you got to make sure you have like a solid foundation, right?
Exactly. You don't want to spend years developing a drug that's aimed at the wrong thing.
And you've brought in some really interesting research on this.
Oh, yeah. From like classic lab techniques
It's an AI.
to AI, exactly. And all of it boils down to this one key question. How do scientists confirm that a specific molecule is actually involved in a disease?
Right, because it's not enough to just kind of have a hunch.
No, not at all. You need evidence.
Hard evidence.
Hard evidence to avoid wasting time and resources.
So let's start with the traditional methods.
Okay.
I'm seeing kind of like two main approaches here, genetic and chemical.
Yeah, those are the two big ones.
So genetic methods,
So genetic methods involve manipulating genes to see what happens.
like actually like tweaking the blueprint of a cell.
Yeah, think of it like you're an engineer and you're tinkering with the design. So one of the classic techniques is the knockout study,
Ah, the knockout study.
where scientists essentially silence a gene.
So they just like turn it off.
They turn it off and then they see what happens.
And from that, they could tell what that gene was doing.
Exactly. It's like removing a cog from a machine.
Hmm.
You take out a specific gene and you observe what stops working.
Okay, so it helps you pinpoint what goes wrong in a disease when that gene isn't functioning properly.
Right. Our sources talk about PEP2 knockout mouse models.
PEP2.
Yeah, they've been really helpful in understanding how the PEP2 transporter affects drug absorption.
Interesting. So like how well a drug gets into the body.
Precisely.
Okay, but I imagine there are limitations to these genetic techniques.
Oh, absolutely. Even with powerful tools like CRISPR, there's always a chance of what we call off-target effects.
Off target.
Off-target effects, it's like you're trying to edit one specific gene,
Okay.
but you accidentally end up editing another gene.
Oh, so you get like misleading results.
Exactly. So you always have to be careful with the interpretation.
It's like needing a double check system just to make sure you didn't accidentally delete something important.
Exactly. Now let's move on to chemical methods.
Chemical methods.
These use something called tool compounds, which are basically small molecules
Okay.
designed to interact with the target of interest.
So each tool compound is like designed for a specific target.
Yeah, like a key fitting into a lock.
Key and a lock. Okay.
So by observing the effects of blocking or activating the target with these keys, researchers can kind of tease out its role in the disease.
It's like seeing what happens when you throw a wrench into the machinery.
Precisely. And, tool compounds are also used in lead optimization.
Lead optimization. So that's where researchers are actually fine-tuning promising drug candidates.
Right. They're assessing things like metabolic stability.
Metabolic stability.
How quickly a compound gets broken down in the body.
Okay, so does it hang around long enough to do its job or
Exactly.
does it disappear?
Right. If it disappears too quickly, there's no point in developing it further.
Makes sense. So these tool compounds, do they always behave perfectly?
Sadly, no.
No.
Sometimes it can be a little bit tricky.
Tricky how so?
Well, sometimes they interact with other molecules besides the intended target.
Okay.
And that can lead to false positives.
So it's like the key fitting into the wrong lock.
Exactly.
Yeah.
You might think you found the right target, but you're actually looking at something else entirely.
So how do researchers avoid getting tricked by these false positives?
It all comes back to rigorous experimentation, using multiple methods to really confirm a target's role.
Okay, so it's like gathering evidence from different sources.
Exactly. You want to build a really strong case.
Yeah, you don't want to jump to conclusions.
No, definitely not. And that's actually a perfect segue into the world of AI-driven target validation.
Ooh, now we're talking. I've heard so much about how AI is revolutionizing drug discovery.
It's pretty amazing. Imagine having this superpowered research assistant
Okay.
that can analyze massive data sets.
Like a supercomputer.
Way faster than any human could.
And what kind of data are we talking about here?
Oh, everything, genomic information, protein structures, scientific literature, all of it.
Wow. And so AI can sift through all of that and find potential targets.
Exactly. And it can even predict how drugs might interact with those targets.
So could it actually help us avoid those accidental delete moments we talked about earlier?
In a way, yes. AI can flag potential issues early on.
That's incredible. It must save researchers so much time and effort.
Oh, absolutely. Our sources mention Atomwise.
Atomwise.
Yeah, and their AtomNet platform.
AtomNet.
It's a really great example of how AI is changing the game.
So how does it work?
Well, AtomNet uses deep learning to sift through enormous chemical libraries,
Okay.
and it looks for molecules that could bind to specific targets.
So it's like a matchmaking service for drugs and targets.
Exactly, and they've even used it to identify potential drug candidates for neglected tropical diseases.
Oh, wow. So AI is giving these diseases a fighting chance.
It really is. It's a really exciting development.
But even with all of his promise, AI in drug discovery is still in its early stages, right?
It's right.
So we still need those traditional methods for validation.
Absolutely. It's about using the best of both worlds.
I like that, the best of both worlds.
AI can provide really powerful insights
Yeah,
and accelerate the process.
but we still need that rigorous experimental validation.
Yeah,
Well,
exactly, to confirm those findings and make sure we're on the right track.
this is all so fascinating. It really highlights the complexity and the ingenuity involved in drug development.
Absolutely. And we've only just scratched the surface.
Oh, I bet there's so much more to uncover.
Oh, yeah. Maybe we should pause here, let our listeners absorb all this.
That's a good idea.
Hmm.
Take a break, and we'll be right back.
Okay, so we're back. And we were just talking about how AI is really like shaking things up in the world of target validation.
Yeah, it's really exciting to see how fast things are moving.
Yeah, it is. But, even AI can't solve every problem, right?
That's right. It can help us find those targets, but doesn't mean drug development is suddenly easy, right? Like there are still challenges.
Big challenges. And one of the biggest is what happens during the metabolism process.
Oh, yeah, you're talking about those reactive metabolites.
Exactly. Even when a drug binds perfectly to its target, things can still go wrong.
And these metabolites, they can cause some serious problems, right?
Oh, yeah. From drug reactions to liver damage, even DNA damage.
That's scary. So it's not just about finding the right target
No, it's also about what happens to the drug once it's in the body.
and like how it breaks down.
Yeah, exactly. Researchers need to figure out if a drug is going to be metabolized into something harmful.
That sounds like a super complicated puzzle.
It is. It's like trying to predict all the possible moves in a chess game.
So how do scientists even begin to predict and mitigate these risks?
Well, there are a few different strategies. One approach is to design drugs that are less likely to form those reactive metabolites.
So it's like designing drugs that are more body-friendly.
Exactly. And AI can actually play a role here, too.
Really?
Yeah, some AI algorithms can actually predict the metabolic fate of a drug candidate.
Wow, so it's like having a sneak peek into the future.
That's a great way to put it. It helps researchers identify potential problems early on.
But even with AI, I'm guessing scientists still need to do a lot of real-world testing.
Oh, absolutely. Prediction is just the first step. They use a combination of in vitro and in vivo models.
Okay, what does that mean?
So in vitro means they're testing the drug outside of a living organism, like in a test tube.
Okay,
And in vivo means they're testing it in a living organism, like an animal model.
got it. So they're basically testing it at different levels of complexity.
Exactly. It's like a dress rehearsal before the main performance.
Yeah, you want to make sure everything works properly before you test it on human.
Exactly. And speaking of complexity, we haven't even talked about protein interactions.
Oh, yeah, those are super important too, right?
Absolutely. Remember, proteins rarely act alone. They're constantly interacting with each other.
They're like little social butterflies.
Yeah, exactly. They form these intricate networks that influence cellular behavior.
So even if a drug hits its intended target perfectly, it could still trigger unintended consequences elsewhere in the network.
Exactly. It's like throwing a pebble into a pond.
Oh, yeah, the ripples spread out,
Exactly.
affecting the whole ecosystem.
So understanding those protein interactions is really crucial.
So how do scientists go about mapping out these intricate protein networks?
Well, they use a combination of experimental techniques and computational approaches. One common method is called yeast-2 hybrid screening.
Yeast-2 hybrid screening. I remember reading about that.
Yeah, it helps scientists figure out which proteins interact with each other.
So it's like solving a cellular puzzle.
Exactly. And there are other techniques too, like co-immunoprecipitation and mass spectrometry.
They all sound so complicated.
They can be, but they all help identify the different protein partners that interact with a target.
But I bet all of that data can be really overwhelming.
Oh, it definitely is. That's where computational biology comes in.
Okay, so how does that help?
Well, scientists use sophisticated algorithms to analyze these huge data sets and create detailed maps of the molecular networks.
So it's like having a Google Maps for the cellular world.
Exactly. And those maps can show how a drug might affect not just its target, but also its target's friends and their friends and so on.
It's like a chain reaction.
Exactly. And it helps researchers understand the bigger picture and anticipate potential ripple effects.
It's pretty amazing to realize that cells are such complex ecosystems.
It is, and it highlights why drug development is such a challenging endeavor.
But as we learn more about these cellular processes, we get closer to developing safer and more effective therapies.
Absolutely. So where do we go from here? What does the future hold for target validation?
Yeah, what's next?
I think we're going to see even more integration of AI and computational biology into every stage of drug development.
So those technologies are going to become even more powerful.
Exactly. And that means scientists can predict and prioritize targets with even greater accuracy.
So it's all about working smarter, not harder.
Precisely.
Right.
And we're also seeing the rise of new approaches like phenotypic screening.
Phenotypic screening that rings a bell.
Yeah, it's a different way of looking at target validation.
So remind me, how does that work again?
So instead of starting with a specific target in mind,
Okay.
you start by observing the effects of a drug on cells or organisms.
So you're kind of working backwards.
Exactly. You see a desired effect, and then you try to figure out how it happened.
It's like being a detective searching for clues.
That's a great analogy. And you know what? Phenotypic screening has actually led to the discovery of drugs that are on the market today.
Really? So it's a proven approach.
It is. It's been really valuable, especially when those traditional target-based approaches have hit a wall.
It's really encouraging to know that scientists are always exploring new ways to solve these complex puzzles.
It is, and I think we're just scratching the surface of what's possible.
Yeah, as our understanding of cellular processes grows, so too will our ability to develop even more effective and targeted therapies.
Absolutely.
All right, so we're back, and we've been on this deep dive into target validation.
And it's definitely been a journey, right?
It has. It's clear that this is like a really critical step in drug discovery.
Yeah, you could say it's the foundation of the whole process.
Yeah, like you were saying before, if you're building a skyscraper, you got to make sure that foundation is solid.
Exactly. You don't want to cut corners when it comes to target validation.
I mean, we've seen how researchers are using all these different tools to do it.
From traditional lab techniques to AI to, a deep understanding of how cells work.
It's really amazing to see how it all comes together.
It is. It's a really cool blend of science and technology.
But I think throughout our conversation, there's been this one theme that keeps coming up.
Oh, yeah. What's
The
up?
importance of context.
Context. Okay.
It's not enough to just find a target and hit it with a drug.
Right. It's like you need to see the bigger picture.
Exactly. Like, what's the target's role in the cell? How does it interact with other molecules? And even, like, how might different patients respond differently?
Right. Because everyone's different.
Yeah, exactly.
So it's about appreciating the complexity of biology and understanding that there's rarely a simple solution.
Right, like those protein networks we talked about.
Oh, yeah. Those are great examples.
They're so intricate and interconnected.
Yeah, disrupting those connections can have like a domino effect.
Even if a drug is doing what it's supposed to be doing.
Exactly. And that's why researchers are really starting to focus on systems biology.
Systems biology.
Yeah, it's this holistic approach that considers like the whole system.
Okay, so instead of looking at just one protein, you're looking at the entire network.
Exactly. It's like the difference between looking at a single tree and then stepping back and seeing the whole forest.
That's a great analogy. So systems biology helps us see how a drug might affect not just its target, but also like everything around it.
Right. It allows us to anticipate potential side effects and develop therapies that are more targeted.
And speaking of targeted therapies, we also talked about personalized medicine.
Oh, yeah, that's super exciting.
It's like the idea that treatments can be tailored to each individual patient.
Exactly, based on their genetic makeup.
So we can predict how they might respond to a drug or even identify potential risks.
It's like moving away from this one-size-fits-all approach to medicine.
Yeah, and creating treatments that are truly customized.
Right. It's about finding the right drug for the right patient.
This has been a really eye-opening conversation. We've covered so much ground.
We have. It's been great.
But I think as we wrap up our deep dive into target validation, there's one question I keep coming back to.
Okay, what's that?
What does all of this mean for like the average person?
Right. Why should they care about this really complex science?
Exactly.
Well, the answer is actually pretty simple. Target validation impacts all of our lives.
How so?
Think about all the medications we take every day.
For everything.
For everything, headaches, heart disease, infections, you name it.
So those scientists working in labs, they're the ones who make those medications possible.
Exactly. They're the unsung heroes of our health.
It's a powerful reminder that science isn't just about knowledge,
No, it's about making a difference in the world.
and target validation is a huge part of that.
It really is. It's about developing drugs that are more effective, safer, and more personalized.
It's about taking those scientific discoveries and turning them into real benefits for people.
Absolutely.
Well, this has been an absolutely fascinating deep dive. I've learned so much.
Me too. It's been a pleasure.
And for our listeners who want to learn even more, we've got links to some of the research articles we've discussed in the show notes.
Yeah, definitely check those out if you're interested in learning more about this really important topic.
And with that, we'll wrap up this episode of The Deep Dive. Thanks for joining us.
And until next time, stay curious.